Then this is another view of the transition, or we say the transition matrix, the rate of transition. But this is from mitochondria. So mitochondria, we spend a lot of time studying mitochondria. I think mostly is because it's very easy to be analyzed in the early days. So a lot of study in the 80s, we do we analyze the mitochondria sequences, and it's very in most of the vertebrate and invertebrates, they are very small compared to plants. So in animals, most of the mitochondria are small molecules, and they replicate in the cell. We inherit that to our descendants. So we use that as the molecular material to analyze the substitution rate. So there's a rich, like sort of a large amount of data or analysis based on mitochondria DNA data. Just as in plants, we do most of the time we work on chloroplasts. That's very similar reason. And for the mitochondria, you can see the mutation rate. So here is like the same thing from one nucleotide state to the other nucleotide states, and we separate all four different sites, different possible nucleotides. Then you have all the combinations, and we try to analyze the pattern we observe from coding sequence of the human mitochondria genes. And you can see one thing obvious here is the differences might be different between the two strains, because you see very dramatic patterning differences. And another thing you also probably observe is there's a lot of changes happen in certain composition, right? So we can see, for example, the A to T change is 0.4, but the A to G change is 14.1. So the pattern is quite different from like the row counts. So you can see the differences in all the mutation directions. So they have put all the possibility here, then you count all the changes from the sequence, and you get this pattern. So you can look at control region of the sequences. So the control region, some people also say it's the D loop region of the mitochondria. And in a lot of the species, they are more AT reached in sequences. So there's more ATs in the region compared to GC. So this is the pattern we observe from the AT reach region.